next generation evaluation system that Mark Young talked about earlier. The person who's leading this project is Benoit Auvray. Benoit, he studied animal science and genetics in Belgium. Master's thesis spent at University of Georgia, writing a genetic evaluation module to analyse categorically and continuous traits together, still used by some in the US beef cattle breed associations to get calving ease EVBs. He set up the dairy cattle genetic evaluation using test day model in French-speaking region of Belgium, one of the two national dairy cattle genetic evaluations. 11 years at Ag Research working for Ovita, which include development of Sheep 5K and genomic selection pipeline. Now at the Maths and Stats Department at the University of Otago, and he is responsible for the Beef and Lamb Genetics New GE project. Benoit, thank you.
Today I'll talk briefly about the results that we have for after a year of work on developing the new SIL genetic engine. My first two slides is going to be a repeat basically of what Mark and Michael have said earlier today. So why do we want to change the genetic engine? And then I'll explain what we've done. So basically we want to develop a new SIL genetic engine that will estimate genetic merit of animals by combining pedigree, the performance, and the DNA information in one big seamless evaluation. And the goal at this point is to have a single weekly national run that will remove the need for all those smaller within flocks and within groups evaluations, and to produce more accurate breeding values. So this is a slide about what Mark's been talking about. So at the top we've got the current SIL genetic engine. You've got all those arrows just denoting different evaluations there. So single flocks plus outside sires, and then small flock groups and larger ones. And then at the bottom you've got the big SIL-ACE evaluation, which is the largest one that runs at the moment with 350 flocks. And that's about 70% of SIL data, and that's run every 2 months. And so those things go into the genetic engine, and then they're reported with like several sets of BVs that go into the SIL BV database. And on the side there's another box that just does the genomic selection now, that uses DNA from some genotyping labs and coming through Zoetis to genomes at the moment, most of them, and EBVs from SIL, and the GBVs are estimated and pushed back into the SIL database. And so in the future, we would only have one evaluation that uses all SIL flocks from a SIL master database, using DNA from a new SIL DNA database that's going to be created. And all that goes into a new genetic engine that creates GBVs directly, and those ones are pushed into the SIL database, and they're directly across flock and can be compared with every other GBV coming out of the system. Now we'll use all of SIL data, and it will be run weekly. So after a year, we have a prototype genetic engine that has been implemented based on some software from the University of Georgia, and those guys are one of the leading groups in computational methods for animal improvement, and they're the inventor of that method called single step that allows us to seamlessly integrate DNA with performance data. And we've run multiple test runs on a data set that's 3 million animals in pedigree and up to 2.2 million records for weaning weight. 3 traits together. So it's a bit smaller than SIL-ACE, but it's not that small. It's actually quite a big evaluation. And we're using 8,600 animal genotypes, which is about half of what we have for the 50K this year, and probably a third to a quarter of what we'll have by the time this system is finished. And at the moment it runs faster than the current SIL system, though. In a few hours we get GBVs there. And the preliminary results suggest that we get an improvement in accuracy for young animals up to 50%. So if you've got a young animal with an accuracy of, for a trait that's reasonably heritable, at 30% accuracy of your EBV now, you might, using this new system, get up to 45% accuracy. And if you've got an accuracy for a trait that's less heritable, say number of lambs born or something, you'd get an accuracy EBV for a young animal of 16%, and then it goes up to 24% using that new system. So for this year, we're planning on keeping fine-tuning the model, and especially for multi-breed. So the structure of the sheep industry is, as you all know, is lots of breed together, and those models might not exactly be suited for that. So there's a bit of work to be done in there so that all breeds can actually be analyzed together. We'll work on a method to calculate the accuracies. At the moment, accuracies coming out of this system are a bit overestimated because they were created for simply pedigree information, not using DNA. And we'll research, so link back to the sheep genomics program that Shannon's leading, to include major genes that they can find on their HD SNP chip to increase the GBV accuracies even further. And then all that needs to be automated and linked with the SIL animal and the new DNA database. And at the same time, we've got a team looking at alternative methods. So Fiona Healy from Abacus and people at the University of Otago, including myself, with some collaborators from Iowa State University and universities in California, are looking at alternative methods to make sure that if in the course of the program, if something better comes up than the system we have at the moment, we're positioned to be able to deliver the state-of-the-art system, so the best that is used worldwide for genetic evaluation.